Hello, this talk is about the natural scenes dataset. This is a 7T Fmri dataset that has been recently completed, and these data are all publicly available. This talk is not really about the content of the dataset. This is since that's been formally described in the paper. Currently, it's posted as a preprint on bioRxiv, but the published version will be coming soon. Rather, this talk is about orienting you to the files and various resources that we have available online to help guide you through accessing the data and understanding what's there. So let's start off with just some of the websites. This is sort of the entry point, naturalscenesdataset.org. It just has some basic information, and you can read this at your leisure. More informative would be other websites. So this is the Amazon S3 site. The Nsd data is hosted on Amazon S3 for fast cloud access. And if you use Amazon S3's website, it sort of looks like this. Here's the directory structure. We'll be browsing the structure in a different way in a second, but I just wanted to show you this just so you understand that it's there. A different website that is relevant is this data manual. This is what's called the Nsd Data Manual, and you get access to this manual once you fill out the data access form. It's available for browsing as a website, as you can see here. It's also available as a convenient little PDF, which is really just a printout of the data manual to a single file, as you can see here. And this is useful, for example, if you want to do a quick find query on keywords contained in this data manual. Anyway, you can access it either way. The data manual will be updated as any issues or things need to get clarified, but currently it is complete as so far as we understand it. But please let us know if it is incomplete or if there are some questions. If there are questions, we can certainly update it, either the main pages or in the FAQ at the end. One last website, I believe, or actually two, sorry. This is an OSF website attached to the paper. It primarily contains these sort of large videos that are cumbersome to put in an actual traditional paper. These videos are accessible on this OSF site. As you see here, I just clicked on Files. They're also in the actual contents of the Nsd dataset, and we will see that later on in this talk. But you can see here that you can just click on these links and watch some of these videos here. That's the primary purpose of this sort of satellite OSF website. And yeah, basic information is just posted here. And maybe one last website I'll show you is, this is an alternative way to browse the contents of the Nsd data on Amazon S3. This is a very lightweight browser-based, essentially JavaScript method. And as you can see here, I can just click around and sort of quickly browse the directory tree and potentially even access and download files. Anyway, we will be using a client, a graphical client to browse, so I will just close this window now. Other primary things, again, I guess I mentioned the preprint. This has all the details on the methods and conceptual stuff and the motivation of this data set, and I encourage you to check it out at your leisure. Okay, so that gives you the basic orientation to what is going on. Just to recap, I mean, the primary sources of information are the paper itself and the data manual. Now, the rest of this talk is going to be giving you a crash course in the data files that we've made available and sort of what is where and how we've organized things. So I'm going to switch over to this little client. This is a little graphical client that uses the S3 protocols so we can connect to the Amazon servers and access the natural scenes dataset bucket, so it's called. And as you can see here, well, this is that just lightweight JavaScript browser index. It's not really that important unless you use it. And we have several directories, as you can see here. I'll be going through this little hierarchy, highlighting key files. I'm going to save the folder called Nsd Data till the end, since it's actually most informative, so it's easy to get these out of the way first. So let's just start with Nsd_betas. So this contains essentially the data set after all of the pre-processing that we've applied. We've taken the raw Fmri data, done basic pre-processing, and then fit the time series of the Fmri data to try to extract single trial response amplitudes for all voxels. And that's convenient because, well, there's some specific ways we do that to try to optimize the quality of these single trial betas, and it just makes life easier for subsequent analysis. I'm just going to navigate down here through, there's 8 subjects, so we're just going to look at subject 1's data. Now, the Fmri data are prepared as a matter of our processing in various spaces, including group spaces like fsaverage and MNI, as well as subject native volumetric spaces. There's 2 versions of that. One's a more standard processing 1.8 millimeter resolution, given the acquisition was 1.8. And we also have a higher resolution preparation at 1 millimeter. So these are volume oriented in the subject native space. And we also have, for your convenience, put the data and transfer this to the subject native free surfer cortical surface, and that's contained in this directory. So let's just start easy with the volumetric 1.8. And if we look in here, there's additional folders. If you're familiar with the data paper, there are 3 versions of our GLM processing. We just abbreviate them B1, B2, B3. So B1 is this beta's assume href, which basically means assume a canonical href when analyzing the data and do nothing extra. So it's very bare bones. B2 has this optimization procedure to find a better fitting href for each voxel, and then B3 not only does that, but also introduces some data-driven denoising through GLM denoise, as well as this ridge regression regularization procedure to better estimate the single trial betas. So we have 3 versions of the Nsd responses, so to speak. Also, if you're familiar, these are not publicly released. These are some auxiliary experiments conducted after the Nsd data set, and so we're not really going to cover those things in this little video. And then I'll just mention finally, the resting state data were also analyzed using a GLM, although it is a bit strange because there's no actual stimuli happening in the resting state other than a fixation cross. But it may serve useful for some comparison purposes. So the resting state data were also fit, and you can look for details in the data paper regarding how that was done. Anyway, we're going to focus on probably what's most interesting, which is, let's just go to beta version 3, and finally we get some of the data files. Now, there's a lot of files, and these are all documented in the data manual. At a bird's eye view, these are the 40 sessions. So these are 40 visits to the scanner. Each visit involves, for the main Nsd experiment, 12 runs of 5 minutes each. So it's an hour of actual functional data. And these are the extracted single trial beta weights for each of the 40 sessions. One side note while I'm at it, we are withholding the release of the last 3 sessions of data from each subject in order to, this is for the purposes of the Algonauts prediction challenge. If the competition is about predicting responses, obviously you don't want people to see the responses they're trying to predict. And so just a heads up about that. 3 sessions, the last 3 core Nsd scan sessions from each subject are not downloadable, although you can see that it is listable here, but it will give you an error if you try to actually download the data. Anyway, so these are the single trial betas. Notice the files are quite large. They come in 2 different versions. There's a sort of a traditional gzip nifty version, which is small, given that it's gzipped. Voxels outside the brain have been zeroed out, so that gives you some space savings. They're also provided in Hdfi format, which is convenient for very fast access, but they're equivalent. Okay, so the directories also contain many other quantities. They're probably not terribly relevant, and they are documented on the data manual, as you can, and there's many files. Just bear with me here. And maybe I'll just highlight maybe a few other files of primary importance. This is the noise ceiling signal to noise metric that we describe in the data paper, and we will look at that in a second. And these R² files are sort of convenient just to get a sense of where signals are. This is simply variance explained by a very simple, what we call on-off GLM model. So the model just says, for each stimulus presentation, assume that there's some sort of fixed level of response. How well does that model explain variance in the time series. So that's sort of a useful quantity, and we'll look at that in a second. Yeah, so that basically covers it. Again, details of all these other files you can find in the data manual. So first things first, let's do some simple inspections. I've already downloaded, just for speed purposes, from this subject 1, 1.8 millimeter subject native space, volume space, and looking at beta version 3, I've already downloaded this r²_session1. So the very first NSD core scan session, this is the essential volume of percent variance explained by this on-off GLM. So just playing with the color map a little bit, we can just use a hot color map and fix the color range a little bit just to see more detail here. Okay, so clearly this is, well, if you know your anatomy, this is pretty sane. This is anterior, posterior, right, left, and superior, inferior. You can kind of see the brain outline here, although it's not obviously very visible. And as you can see here, it looks fairly sensible that we're getting very, very strong signals in occipital cortex. So this just an example of what you can do. You can easily load it into some viewer that knows how to load Nsd files. Let me also load in this noise ceiling signal to noise volume here. Here, I guess you can see the brain structure much more clearly. The meaning of the quantitative values, again, refer to the data manual and paper to sort of source that out, suss that out. But here we can just do some quick visualization just to get some understanding of the structure of the data. Of course, we can toggle between these two images, and clearly they're highly related, and that's expected. Okay, again, this was just from the very first scan session, and there's up to 40 for this this one subject 1. Now I'll just turn to one example of the Hdfi file. We could, in theory, try to load the Nsd. It is quite large and cumbersome. My understanding is you may have to unzip every time you want to load it from one of these gzip files, which might be annoying. Here I've already downloaded the Hdfi file, which is 1 GB for all the single trial estimates obtained in session 1. And what we're going to do is just quickly load in these data. Actually, this may not be quick. It's trying to load 1 GB of data in this one line here. So I'll just let that load. And in the meantime, I'll explain this next line. So one of the advantages of the Hdfi file is it's saved as just a binary file, so you can randomly seek different parts of the file, and depending on how it's saved, this can provide massive speed improvements. And so in this little example code here, I am just pulling out a small little window of 20 by 20 voxels, starting at the 30th and the first voxel in the first 2 dimensions. And from the 40th slice, I'm just pulling out one, little square of voxels, and I'm getting all 750 single trial betas. And you'll see in a second here, once this is finished, that this can execute in very fast, sub-second. And this is because you can randomly seek in this file. So it's loading. Takes a little while. In the meantime, what I can also show you, oh, it's done. Let's go ahead and do that. So let's just check some basic characteristics. Remember, this is the subject native volume space, so it has some idiosyncratic numbers for the 3D dimensions of the volume. And every Nsd core session has 750 trials, like actual trials where the subject sees an image and makes a response, and that explains that dimension. Now, one thing to be aware is, for space reasons, the data, the betas are saved typically, although check the data manual for full details, in an integer format, and this just cuts down on the storage space. And so you have to be very careful to convert it to a decimal format, like double here, in MATLAB speak at least, to do some actual computation. So at a glance, I guess, we've loaded in sort of the end of our processing that we prepare for the Nsd data. We have, for every voxel, 750 single trial beta estimates. And remember, this is pulled from the B3 preparation, this one. So let's just look at some of the data just to get a sense. So here's a line of code that is very simple. It just pulls out the 40th slice out of 83 slices, converts it to double so we can do computation on it, and all it's doing, oh, one last thing, sorry, I should mention, when saving in integer format, the units of the betas are intended to be in percent bold signal change, but as most of you are probably aware, Fmri bold signal change is very small, sometimes as small as 1 or 2% or 3%. And what we've done is before we save into this integer file, we multiply by 300 just to ensure we have sufficient dynamic range. So as soon as I load from this beta file, I divide out by 300, which puts us back in percent signal change units. So that's what I've done here, and I've just simply taken the mean across all trials. So if we go ahead and do that, so again, one slice, looking at mean percent signal change across all of the single trials in this very first scan session, it looks something like this. You can tell this is an axial slice, and the ordering is LPI, so L is first, so the left is the beginning of the first dimension, and P is posterior, so the beginning of the second dimension, as you can see here. And the third dimension is I, meaning the first slice is inferior, and then it traverses upward to superior as you move from 1 to 83 here. Anyway, so this looks pretty sane. And just as sake of an example, I mentioned earlier, what if we wanted to just zoom in and isolate a small section of voxels? So here again, I'm pulling out a 20 by 20 square of voxels from the 40th slice, and this can execute very quickly. As you can see, it's already done. And just to confirm, we've got 20 by 20 voxels. We only have one voxel, I guess, in the third dimension, and we have all trials. So similarly, we can visualize these data, again, remembering to convert to percent signal change like so. And as you can see here, not sure why that didn't run, but this is just a small window, 20 by 20, from the data here, as you can see the correspondence. Okay, so that's just a quick introduction. Obviously, there are different ways to load in Hdfi files if you're using Python or other tools. Okay, so this was all pulled from the 1.8 millimeter subject native volume preparation. Now, if you look in this other folder, functional 1 millimeter, this is the high-res preparation prepared using some upsampling, which has some benefits, we believe. And the files and everything are comparable. So here I've just opened up a folder from the 1 millimeter, and you can see all the usual files. Now, for convenience, I've already downloaded one of them, and I was just going to pull that up just for comparison. So here's the sort of on-off R² from the first session of the 1 millimeter preparation, and I've kept open the 1.8 millimeter. So here I'll just do some quick, let's make everything comparable in terms of the color maps. Let's just do 0 to 50. Actually, that's too much, or too little. Let's do 0 to 75 for that one, and then we can do the same for the 1 millimeter preparation. So that's 0 to 75. Let's make it both hot color map, and just sort of show that things are consistent. The pixel size, of course, is different across these two preparations, and there's some slight technical details on exactly how you can think about the spaces that these two data sets live in. But as you can see, things look pretty sane, at least at a bird's eye view here. Okay. The 1 millimeter preparation, I should mention, is quite large, so it's a bit cumbersome to work with, but if your scientific question really relies on getting as precise and high resolution as you can, you may want to consider using it. Okay, so popping back up a few levels here. So we've just been in the Nsd Data betas directory. So this is, in a sense, the largest, and it's where the bulk of the data lie. So if you're willing to use our single trial beta estimates, those are quite large still, and they're all sort of stuck in this one directory just to keep things nice and segregated. Now I'm just going to move on to this next folder. So if you're interested in the raw imaging data, they're contained in this folder called Nsd Data _ raw data. The files and directory structure are formatted in BIDS format for your convenience. And so things should be fairly straightforward. I'm just going to show you an example. So navigating the tree down to subject 1, we have the 40 core Nsd scan sessions. We also have the anatomy stuck in its own folder, Nsd Anat. aside for diffusion, which is stuck in its own folder. Remember, these are the auxiliary experiments that are not described nor released yet. And then finally, we have the initial 70 screening session, which actually involves localizer experiments called PRF, which is a retinotopic mapping or population receptive field mapping experiment, and FLOC, which is the functional localizer experiment, which is for finding category selective regions. Now, I'm just going to show you an example of what's in the first core Nsd session. We did collect field maps. There were several field maps. This is a dual echo sort of gradient recalled field map style of field map. They exist here, and you can check that out. But I'm just going to focus on the functional data here. So in the first scan session, the first Nsd core scan session, there are 12 runs, as you can kind of see here, 1 through 12. And this is all sort of formatted according to bids. And just for fun, we're going to look at the first run. So that would be, I guess, this Nsd file run 1 bold. I've already downloaded that here, so we can let ITK Snap try to load that. I will also show, while that's loading, I guess the first T1 anatomy that we acquired for this subject. That's contained in sesh Nsd anat. So if you navigate down there, you see the T1. There's actually other anatomies. So we also got several T2 acquisitions, as you can see here. We also got a high-res T2 weighted scan for segmenting the hippocampus. We also got venogram and angiogram. But for this video, I'm just going to show you the very first T1 we acquired. Notice there are, for this subject, up to 6 repeats of the T1, just to allow averaging to improve the signal to noise in the T1 even further. I've already downloaded this first T1 weighted volume, as you can see here, and we will go ahead and open that up in my second little session of ITK Snap. Okay, so this is just raw data. Well, raw, at least in the reconstructed image sense from the scanner. It's been dephased, as you can see. And obviously you can see white and gray matter. Things look pretty okay, and yeah, that's really all there is to say here. Now, finally, our raw Nifty from the very first run and the very first subject of the first Nsd core session is here. So again, this is just raw data. One thing we can do is do a quick scrubbing through time just to get a sense of temporal stability and head motion and all that. And subject 1 was definitely one of the rock solid head motion subjects in a sense, and as you can see as I scroll, there's barely a voxel shift, at least in this very first round. I mean, there's a little bit if you in sort of a rocking back and forth session, as you can see. Anyway, so this is the raw data, and you can obviously visualize it and think about the quality. There's dropout in the usual locations near the ear canals, and obviously distortion near the frontal orbital frontal cortex. This is all expected for Fmri data, at least acquired using API and gradient echo. Yeah, okay, I think that's all I wanted to show here. Let's just close that up. So again, we were just browsing the raw data prepared in Nifty format for subject 1. Okay, so let's close up this directory tree here. I will mention this directory. The Nsd Data Stimuli is an entire folder just dedicated to the natural scenes that we were showing in the experiment. There's actually only one, at least in terms of the Nsd core experiment, one file in this directory, and that's this Hdfi file. As you can see, it's a very large file, almost 40 gigabytes. It's pretty straightforward. The resolution is 425 pixels, so it's 425 by 425 by 3 for the RGB channels by 73,000. And this is because we prepared 73,000 images. These were taken from the Coco, Microsoft Coco image database. But we took them and did some light selection and cropping, and it's all been prepared in a single gigantic 4-dimensional matrix and saved in an Hdfi file. Again, the primary advantage being you can very quickly load individual images from this single binary file. Details on how to manage that file are provided in the data manual. last, we're going to look at time series, and then we will move on to the sort of main folder of interest, the Nsd data folder. But let's do the time series. This folder contains what you might call basic preprocessed Fmri data. So there's still time series and head motion and distortion and slice time correction, and all of those things have been taken care of. So these are sort of a nice starting point for certain types of analyses, especially if you're interested in resting state. Typically, people need to work with the resting state time series. So I'm going to drill down here and look at subject 1. There's actually multiple things other than imaging. The eye tracking data is actually stored here. These are the raw EDF files from the iLink that we used. Eye tracking was performed only in a couple of scan sessions, and they varied depending on the subject, and details are available in the data paper. This is the preprocessed version of the eye tracking data, so we've already done some light preprocessing to deal with the eye tracking data. We also include video captures of the eye tracking computer. This is because the eye tracking was actually quite variable, and it's very hard for various reasons in our 7T scanner to get a good lock on the pupil. So this was basically a backup option. I've already downloaded one of the files here. I forget where it came from, but I don't think it was subject 1, or it might have been. Doesn't matter. But it's just to give you a sample of what these videos are like. So we've already cropped these videos to be exactly the duration of the actual Fmri acquisition. Again, details are in the data manual. And so as I scrub through this video, in this case, the pupil lock was actually quite good, as you can see down on the left, and you can see the XY traces here. So this is actually an example of good eye tracking. But certainly many sessions did not have good pupil locks, but these videos might be useful just to get sort of ground truth on what was actually happening in the scanner. Anyway, Physio Data, so this includes pulse oximetry and respiratory belt for the subjects. These were acquired during the 10 sessions in which we conducted resting state runs in addition to the Nsd. 2 of the subjects actually got further resting state, but most subjects just got 10 sessions, 10 minutes each. So that's 100 minutes of data. These are just text files of pulse and respiratory, as I mentioned. We'll see inspections of these data later on in this video. But let's just turn to the, probably of primary interest, the actual Fmri time series after basic preprocessing. Just a brief note, the design matrix information is also contained there. These are motion estimates from the rigid body motion estimation that we used in our preprocessing. But the Fmri data themselves are finally in this folder. These are separated by run, and we have the kind of auxiliary experiments here. This is the initial 7T localizer screening session that we ran, and then we get 40 sessions times typically 12 runs, although in the resting state sessions there's 14 runs for this subject, and it goes on and on and on up through session 40, I suppose. Now, I've already downloaded one of these just to as an example. I believe it's here, although it'll take a few minutes to load this, I suppose, on my computer. Okay, so while that's loading, let's sort of navigate back and orient ourselves. So this is, again, this is the preprocessed Fmri time series, which is a precursor to the actual single trial estimate extraction, which is in the betas directory. So this, we're going to briefly look at this preprocess file, but next up on deck is turning our attention to the Nsd data folder. So the Nsd data folder is potentially most useful to have, for example, like a quick access on your local computer, because it contains a lot of figures and sort of the basic anatomy and ROIs and things like that that are lightweight, or at least lightweight compared to the massive single trial betas, or even the massiver preprocessed time series. And I've already downloaded this folder of Nsd data to my local computer. Oop, pause on that. Finally, it's done. So remember, this is the preprocessed time series for an example run in a subject. And this is from the 1.8 millimeter preparation. So as I scrub through the volumes here, you can see that the brain is stable, which is expected since we attempted to make it stable through the entire preprocessing, including motion correction. but you can see it's pretty straightforward. Like the only thing that's been done is some voxel-wise temporal interpolation to correct the slice time and to regrid the time on a slightly finer grid. Details are in the data paper. So in addition to a time interpolation, there's a spatial interpolation to correct head motion and registration and EPI distortion and all that. And so after the result of those two processes is shown in this image here, and it looks like a brain. Looks pretty sane. Okay, so let me navigate back. Right, so the rest of this video is going to be focused on the contents of this last directory called Nsd Data. Okay, so just go one by one. So behavioral data is contained in this folder, and I won't explain all of these little things. This is documented in the data manual, but a variety of various behavioral measures, post-scanning questionnaires, behavioral outcomes of the PRF experiment and the FLOC experiment. So that's that. Experiments contains sort of meta information on what were the experiments actually like. These are calibration and measurements of the monitor we used. This is information on the FLOC experiment. There's a convenient little screen capture that you can just play to get a sense of what was the experiment actually like. This is literally what subjects were seeing in the scanner for the FLOC experiment. Here's the PRF experiment. Here's the resting state experiment, although some people would not call it an experiment, but it is an experiment. It's basically a cross. And then probably most importantly, what was the Nsd experiment actually like? And that's in this screen capture here. I'm just going to skip in the middle here. The images were shown for 3 seconds, and there was a 1 second gap before the next trial. And the subject's task was to do what we call a continuous recognition task. So for each image, you're trying to gauge whether you've seen that image at any point in the scan session or even any of the previous scan sessions. So it's a very long-term memory task, very challenging. And they're making a response as soon as they feel like it on each trial. Okay, so that covers this experiment. Oh, one last thing. The actual MR pulse sequence protocols that we used for the experiment are contained in these PDFs here, so you can look at those in detail. Moving on, I'm going to leave some of these big folders till after, so I'm going to first tell you about information. These are small informational files, demographics, and log and logging on each scan session and things like that. Again, these are documented in the data manual. Stimuli. So if you remember, the Hdfi file of the 73,000 images was contained elsewhere, but these are more convenient versions of that. There are a special subset of 1,000 stimuli that we showed to all 8 subjects, and a quick movie showing you all 1,000 of those special shared images is shown here, just to give you a nice convenient sense of what the Nsd stimuli were like. They're also contained as PNG files here. They're just numbered from 1 to 1,000, and there's also a sort of Nsd 73K ID that we attach to each image. Anyway, so you can also look at the images in that format. I will also mention, I guess, these files. These are showing you the PRF experiment. These are what, at least I call the apertures. So basically just a coding of where the texture-y object thing existed during the experiment. And that's provided for the bar type scans, as you can see here. And we also have one for the wedges and rings scan types. And that's shown in this movie here. So here we have a rotating wedge and also some ring stimulation. Those are useful if you want to analyze the PRF data yourself. We also have analyzed the PRF data and provide results of that in the Nsd data files. And templates, sort of miscellaneous information on various files and information and flags that were used in pre-processing the data. One thing of interest, this is an MNI 1mm template that we take from FSL, and we did use this to do some of the MNI-based registration and things like that. Okay, so now let's turn to sort of the meaty directories. This is a FreeSurfer directory. So we use FreeSurfer to reconstruct the cortical surface and the 8 subjects based on the T1, actually average of the multiple T1s we acquired for each subject, and this was done at 0.8mm resolution, and this was passed through FreeSurfer using the high-res flag to take advantage of that 0.8 acquisition. And we have here just the usual subject directories from FreeSurfer, and we're just going to take a look at subject 1. All the usual FreeSurfer outputs are here, but we also stick in these directories many additional outputs specific to Nsd. So you can see here some of the FLOC analysis results. We have some atlases, cortical parcellations atlases that we've prepared for the subjects. Here are some PRF results. And ROIs actually also exist here. So for example, LH dot, let's see, LH dot, oh, here we go. So LH.PRF-visual ROIs is a collection of visual area ROIs that we've manually defined on basis of the PRF data. So these files exist, again, in these FreeSurfer directories. And now I'm going to show you a quick example of just loading some of these files just to demonstrate that it all works. I'm just going to launch Freeview as like a quick method to visualize some surface data. So we're going to load in for subject 1 the left hemisphere inflated surface. Yep, we're already in subject 1, so let's just go find LH.inflated. So this is the reconstructed cortical surface after puffing it up a little bit. And it should look pretty typical. And we're going to load on this, just as an example, the PRF-visual ROIs file that I just mentioned. So this is the left hemisphere. We can obviously rotate and do all sorts of stuff with it. And so just as an example, I'm going to load in as an overlay here. Label directory, we have, scroll down to LH.PRF-visual ROIs. So this is a discrete integer labeling. I guess we'll see that in a second. So let's just go ahead and load it in. We got to change some of the coloring here. So the values consist of 0, which means unlabeled, and 1 through 7, at least for this file. And you can see in the back of the brain, we have V1, V2, V3, and HV4. So let me just rotate this around a bit. Of course, we don't know which number means what, so there is a text file that helps inform you on that. That file is called, let's see, oh, here we go. So PRF-visual ROIs.mzz.ctab. It's just a text file, so we can just open it up. And it just contains a little text labeling for the 7 regions that were labeled in this ROI file. And so we have V1 ventral, V1 dorsal should be 1 and 2. We can go back and just sanity check that that's true. So V1 ventral should be this orange color, which is orange, which matches the labeling here, and dorsal should be 2, which is 2, which is yellow, and so this is clearly correct because that's the calcarine sulcus. Okay, so let's move on. Quit out of that. Next up, aside from the FreeSurfer directory, let's talk about PP data. So this stands for preprocessed data, in case you were wondering. This contains a lot of sort of the, well, it's not the massive Nsd core betas or time series, but it's anatomy and a lot of sort of auxiliary quantities that are of interest. Let's just start with anatomy. There are a lot of files here. You can, again, get a documentation of it on the data manual. I'll just sort of zoom in on the maybe most important or typical ones. So here's a T1 So for this subject, this is after some pre-processing, and that's what makes it different from the raw T1 that we saw a few minutes ago. As you can see, there's some brain masking happening. This has already been averaged across multiple repetitions, and so on. So this is a 0.8 millimeter resolution. We also create other resolutions that may be of use. We also have a T2, and this has all been co-registered and everything. So if we load this, they should be exactly in register, and you can eyeball check this for yourself if you're curious about that. So we have a T1, T2, we have an angiogram here, we have a venogram, we have a bunch of other files. I will sort of skip over that. Maybe I'll just highlight some ROI files, I suppose, here. So there's a large collection of manually defined and sort of Atlas automatically defined ROIs that we have already generated. Let's just start with a simple example. So LH, actually let's do the same thing we just were staring at a second ago. This is left hemisphere visual ROIs. So we can load that in and confirm that stuff seems coincident. I guess we should change the color map. Let's just do, actually I'll explain this for a second. So there are values -1, 0, and 1 through 7. So -1 actually means outside of cortex, so that would be all of this value out here, as you can kind of see there, although that's probably a rounding error in the visualization software. So that's -1. 0, I believe, corresponds to gray matter, at least as determined through the processing that we applied. And then the rest of the values above 0 are the actual ROI labels, and that's of primary interest. So I'm going to change the color map here and zoom in on that. Okay, so let's just pan a little bit and just get a sense of where things are here. So here's our T1, and here are the ROI labels in the exact same space. These ROIs are in also 0.8 millimeter anatomical space, so they are matched here. I suppose we could do some overlay to try to get a better sense of how well things are matching up here. And things look pretty reasonable, and all the numbers should also work out. So remember, V1V is assigned a value of 1, and so this dark blue color is our indication of where V1 ventral is, and it does look reasonably correct. Okay, so there are many other ROI files that may be useful. Oh, I should just mention LH obviously means left hemisphere, and that is why only the left hemisphere, which is actually on the right-hand side here in ITK Snap, is labeled. A combined ROI file is provided here, which doesn't have the LH prefix, and perhaps, obviously, it will have both hemispheres labeled, as you can see. Whoops, although the other one's visible, let me close it. Okay, there you go. So this ROI file has both hemispheres labeled using the same number for both hemispheres. Okay, so I think that covers that. There are many other ROIs, of course, that you can inspect. Now let's move on to, let me just briefly mention this. This is the Nsd Behavioral Results, and it's a large text file, tab separated, which you can open in a text editor or in your favorite programming language. Here I'm just going to open it up in Excel, and it contains all the basic sort of reaction time, correctness, what stimulus was this, all that type of information for all Nsd core trials conducted for this subject. So this is just subject 1, and we get 30,000 trials. I guess the first row is the header row, that's why it's 30,001. So this is all the, again, behavioral data on the continuous recognition task that we had the subjects perform. Going back to imaging, this is the anatomical data for this subject, and these are the subject native volume spaces that we prepared for this subject. Let's drill down into the 1.8 millimeter volumetric preparation. Again, there's many files. These are sort of results of the FLOC experiment. So we've already pre-processed the FLOC experiment and analyzed it using some sort of flavor of a GLM to get some results out, and those results are contained here. There are a lot of files. The mean is just the mean signal intensity, so actually we should maybe just look at that just to get a sense of what we're talking about. This file is actually the mean of 40 sessions. So we have a mean within each session, and then we actually take the mean across all 40 scan sessions, and that's this image here. It is just the mean intensity of the preprocessed Fmri data. Let me just highlight a few things here. So analysis of the PRF experiment, I guess something easy to look at would be eccentricity. So if we load that in as an additional image here, the ranges are a bit crazy, so you have to be careful about the color map here. Let's just keep it from, whoops, next one. Let's use something reasonable. Okay, it's a little hard to see here, but there should be a progression from foveal to periphery. It's a little hard to see in the volume view, but anyway, clearly there's some structured signals in the back of the brain, and I think that's sufficient for this example. Moving on to the actual Nsd experiment. So I did mention this R-squared thing. So this is the variance explained by on-off GLM, and it's just very useful just to get a sense of where signals are in the Nsd data. So we can load that up, and you can see that it looks at least qualitatively similar to what we were looking at earlier, like so. Okay, so that works there. And we also have, it sort of depends on how you want to structure your analysis, whether you want to do, presumably you want to do your analysis in the functional space, not the anatomical space. And so in the functional space, you also have the full set of ROI files, which can be quite useful. I will load in, just for example, this Cortical Atlas. This is a visual topic or sort of visual area atlas that we took from a previous paper. It is an atlas that's sort of based in FS average space, and what we've done is taken that atlas in FS average and projected it back to the individual subjects, and we then can derive estimates, at least, of where these various areas are, obviously with some level of precision. It's not perfect, but it's a reasonable guess. Now I'll just change some of the color maps here. In this file, there are 25 different ROI labelings, and I can just change some of the color maps here. So I'm just doing it from 0.5 to 25. And what we can do is maybe I can just swap between these two. Okay, there you go. So just get a sense of, this is totally from the atlas, and there are discrepancies. This atlas extends further out in the periphery than our Nsd experiment, so we don't expect everything labeled here to get driven very strongly in the Nsd experiment. So just be aware of the things like that. Oh, and also the atlas doesn't extend all the way to the fovea, whereas the manually defined, V1 through V4 ROIs that we provide do extend all the way to the fovea. Anyway, so if we just toggle between these two, it looks somewhat consistent. Of course, these are tricky issues of how you convert between these atlases and gray matter and all this stuff, but it doesn't look terribly off. Okay, that takes care of that, and maybe I'll just mention, some of these files. So just for convenience, primarily you'll probably be working in the functional volumes, the volumetric space for the functional data. So for convenience, we've already created some versions, depending on what you want to do. This is just the T1 anatomy data that's been mapped to the 1.8 volumetric space for this subject. And it's exactly in register and everything with the functional data. So if I toggle between these two images, as you can see, they are well aligned. The T1 looks a little ugly, of course, since this is a 1.8 millimeter space, and that explains the visual look here. Excuse me. Okay, those are the, I think, primary files of interest here. Let's see, anything else? Nope. Okay, so that covers some of the files that are available in this PP data folder. Lastly, this is probably only for technical uses. These contain all the transforms that we already pre-compute for moving between all the various spaces, including MNI and FS average and the free surfer and the volume and the anatomical space. Details are in the data manual. Typically, you won't need to really interpret or use these. These are used as part of the Nsd map data utility, which we provide with this data set, which allows you to move and resample between different spaces. Okay, so that takes care of that, and I think last but not least, we turn to this inspections folder. These are essentially a bunch of figures and videos and movies. They're useful just to get a sense of the data quality and the scope of what is in this data set. I think I'll go through each of them at least rapidly because they're just pictures. This is what we call the noise ceiling for the B3 beta version. This is averaged across 8 people, so this gives you a sense of where sort of stimulus reliable signals are in the data set as a whole. Again, this has been averaged across 8 people, so you could look at it at an individual subject level, and the averaging was done in FS average space, just so you know. Co-registration is always tricky, so these are movies that give you some nice quick views of how good the co-registration was, at least in our pre-processing that we applied between the T1, T2, and functional data. There are various other movies that you can stare at. This is between the T2 and the very high resolution SWI, i.e. venogram. So these movies exist for your perusal. Eye tracking inspections. So the latest version of the Nsd data paper does include pre-processing and analysis and quantification of the eye tracking results. This is a very large folder of results showing you for every single run plots of the raw and pre-processed eye tracking data. I won't go into details. Certainly some of the data looks quite clean. This is actually plotting the pupil response, so there's a very clear related to the experiment type pupil response. I guess what we're more familiar with are the XY traces. Subjects were asked to keep fixation. Obviously that's challenging for a very long experiment, and we can go in and do some quick inspections of the data for every run and every session that we did attempt to do eye tracking in. Moving on, these are just a sort of a record of how we exactly cut these cortical surfaces to flatten them out. We include flattened versions of the cortical surfaces for each subject, and this was done manually, and we attempted to be consistent in how we did it for each subject. So the record of how that looks is provided in this folder. This shows you a kind of a sanity check that the 8 individual subjects are pretty well aligned with the FS average curvature. So FS average is a group space based on some sort of optimization of the sulci and gyri patterning of the subjects. And here I just show you a movie where I cycle through the 8 subjects, show you the average of the 8 subjects, and then show you the FS average curvature. And you can use this to sort of sanity check that, at least at some scale, this cortical-based alignment looks pretty sane. This shows you some details on how we did the functional to anatomical alignment. In a fine transformation, we did not find to be sufficient to align the 7T preprocessed Fmri data with the 3T T2 weighted image. So you can sort of cycle between these two to confirm that. And so we used a very slightly nonlinear warp, as you can see here. So the difference between this and this is the extra freedom afforded by the nonlinear warp as implemented in ANTS. And so you can confirm for yourself that this was probably a good idea to maximally make a match with the, in this case, the T2 target. So we take the T2 to be the target, which is what we're trying to get at, and you can look at these yourself to kind of see how big this sort of nonlinear adjustment was in order to perform the match. These figures show you a little bit of the size of the gradient nonlinearity effect that we talk about a little bit in the paper. This is sort of how big the effect is at 7T for a sample run. And this is how big the effect is at 3T. It's very, very small at 3T. These are sort of grand bird's eye view of the data set. And this movie in particular was referenced in the paper. This just shows you, after pre-processing, the mean volume from each of the scan sessions that we held for each of the 8 subjects. And you have axial, sagittal, and coronal views. And so basically we're looking for stability here. And certainly there's some amount of visible instability, somewhat more so for some subjects than others, and somewhat more in certain parts of the brain than others. And this is expected given the heavy distortions that occur in frontal cortex and I guess near the ear canals again. But you can use this just to get us, again, qualitative a sense of how well these things are getting handled. Subject A8 is potentially the worst of the 8 subjects due to high levels of head motion for this subject. And there is one little effect that you can kind of see here. There was a sinus infection that persisted for a few weeks, and that shows up very clearly in front of the frontal cortex here. Complementary view of this same idea. Now this is plotted on the FS average surface, and this cycles between each session for each subject. And as you can see, every subject has a very idiosyncratic and very reliable pattern of mean signal intensity. Dropout effects are in the temporal cortex, or near the temporal cortex here, and frontal cortex. The remaining patterning is probably due to vasculature or veins that do show up in the mean intensity in the Fmri data. But I guess the main point of this figure is just a complementary view of what we were just staring at earlier, whereby things are highly stable within a subject, and across subjects there's actually substantial differences. And again, I should mention this is all in FS average space, so these are theoretically aligned cortically or curvature based, and yet there's still massive differences, at least in this mean signal intensity. Similar visualizations for this on-off R-squared metric. So I'll just breeze through this. Some days subjects are better than others. This can be due for a variety of reasons. But again, I guess we're looking for general stability, ensuring that we are, to some degree, measuring from the exact same cortical locations across many, many sessions. And here's the corresponding surface view of that. Again, stable within a subject, and there are actually strong differences, so individual differences, even after curvature-based alignment to FS average. Okay, these are visualizations of the extremely high resolution T2 weighted scan we did for the hippocampal segmentation. I will just skip over that for purposes of time. This is a view of how well the T1 to MNI registration was. I'm just cycling between the 8 subjects that have been warped to MNI, and the last image is the MNI template pulled from FSL. And you can just sort of appreciate for yourself that alignment is pretty good. Of course, the cortex itself is highly folded and is not extremely well aligned using this volume-based nonlinear warp. Head motion is obviously a huge problem in Fmri, and these are just very convenient inspections of, these are the 6 parameter rigid body parameters estimates that we attained in our pre-processing for every run, and the runs are separated by vertical lines. So in a glance, you can look at all the head motion parameters within a session, and we have all 40 sessions for subject 1, for example, here. Subject 1 looks quite good, as you can see. If we scroll on to other, perhaps the worst performing subject was subject 8, and you can see in their motion visualizations, you can already see small transient head motions for that subject. Physiological data inspections are also provided. Again, these were done during the sessions where sessions 21 through 30, where we collected the main body of the resting state data. This is the pulse oximeter. On the whole, the data quality of the physiological data was quite good, with some exceptions. Probably a few of these will start looking strange, so this is probably noise, obviously worth looking into further, but for the most part, the physiological data worked quite well. The respiratory data is a lot lower in frequency, so it'll be clear what the signal we're looking for is in this data. And so as you can see, at least for these sessions, it's extremely clean physiological data. I should mention the very last run In this session, there were 14 runs. The first and last were the resting state runs, whereas the central 12 were the Nsd core experiment. And we asked the subject in the second of the 2 resting state runs to do an instructed breath. And this was triggered by a fixation cross turning red, I believe, about, I think, 12 seconds, but check the paper for details there. They were instructed to take a deep breath, and this can be quite useful for sanity checking the validity of the processing of the physiological data. This is a bit similar to what you saw earlier, except here we're doing something a bit funny. We are basically choosing a random volume from throughout a year of data collected on these subjects, and we're just jumping randomly from time point to time point. And this is sort of a very stringent test of stability, and it highlights potentially worst possible scenario for how well we can believe the Fmri data have been pre-processed in terms of spatial stability. Again, you can see for subject 8, perhaps in frontal cortex, things aren't extremely great. Some subjects are a lot better, so I don't know, subject 1 and 2 probably show higher levels of stability. Certainly there are different ways to pre-process and handle the data, which might improve spatial stability in the future, but this is how it currently looks given the processing that we've performed. These are just very convenient movies of just a sample run from each of the 8 subjects in terms of their raw data. So we've sort of already done this earlier, looking at the raw Nsd from the BIDS formatted data. You can look at these at your leisure, and then pp is not the raw data, but after our basic pre-processing, how do the data look for this exact same run? And so you can look at these to get a better sense of the general image and stability quality here. ROIs. There are a lot of ROIs we provide, and there's a whole collection of figures, and which might be quite convenient to help you get a grapple on what all is available. Cortical Sulc is a FS average oriented anatomical atlas that we've prepared, highlighting a few of the major sulci that we believe are useful to understand where they are. So you can use this if you like. And I'll just highlight a few of these files, figures here. So in the place selective analysis using the FLOC experiment, we defined 3 regions: occipital place area, OPA, perihypocampical place area, PPA, and retrosplenial complex, RSC. I'll just show these images. This is a probabilistic map across the 8 Nsd subjects. So for each subject, we went in and, on basis of the FLOC Fmri data, did some analysis and tried to decide where these regions are in each of the individual subjects. And what we've done here is projected those manually defined ROIs to FS average space, and then plot the fraction of subjects with this particular ROI OPA labeled at each cortical location on the FS average surface. So you can see that there's some consistency in where this lands in FS average space, but obviously some inconsistency given that some of these regions only have a fraction, maybe 1 or 2 of the 8 subjects labeled. And so that's how it looks for OPA, this is how it looks for PPA, and this is how it looks for RSC. One word of caution, the way we define the ROIs was quite liberal, and this is intentional so that you can further restrict the ROI according to some statistical threshold according to your needs. But these were intentionally quite liberal, and so that's why this looks quite large in this view. Moving on. These are not terribly of interest. We actually saw visualizations of this in the movie we played earlier, but this is just the entire volume as a montage showing you the mean signal intensity and the on-off R-squared that we've seen several times already. But might be useful if you want to look in very careful detail at individual sessions and individual voxels. This is a convenient visualization of the entire flattened cortical surface. Actually, there's 2 versions. Let me just orient you here. So there's something called FS average and something called native. So native is a little more straightforward. This is just the idiosyncratic flattened surface that we've cut for each of the 8 subjects, and you can see the outline of the flattened cortex is slightly different across the 8 subjects, and this is expected given individual variability. Now, we can take these data, I mean, in a sense, the curvature are data, and you can take the curvature data and project it to the FS average surface, and so that's what this image is. So as you can see, the outline of the brain surface here is exactly the same across the 8 subjects, because this is, again, the FS average surface. However, the curvature data that's getting plotted is different. So if you zoom in very carefully here, say on the this sulcus here, you can see, if you look carefully, that the exact way it looks is a bit different across the individual subjects. And again, this is something we already saw in sort of a movie format earlier, but this is in a static form. Okay, quality of the cortical free surfaces, and we have here, for your convenience, movies, just scrolling through the T1 with the cortical surface outlined on top of it. So this is subject 5, a sagittal view. This was generated just using Freeview and made into a movie, showing you a visual inspection of the quality of the cortical surface reconstructions that we obtained. We believe it's pretty high quality, depending on your level of tolerance. Certainly some regions of the surface are imperfect, and we've actually labeled them with little blue colorings here, and we provide sort of quantitative files just to alert you to where these locations are. They're typically in the hard regions, like, again, near the ear canal and near the frontal cortex up there. And then finally, we have another folder of figure files. Hopefully these are useful to you. Well, there's a lot of different information in here. Maybe I'll just highlight the main ones. So these are plots of the noise ceiling of the 3 different beta versions that we generated, and we can see a progression from beta version 1 to beta version 2 to beta version 3 using a fixed color map. Obviously these values are getting larger, which is good from the point of view of the noise ceiling metric. There are a lot of other figures here. This is the HCP multimodal parcellation atlas plotted with or without the labels, which gets quite complicated. That might be of interest. Probabilistic maps. So we saw this for PPA or the place selective regions earlier, but there are many different ROIs, and that's all shown in these various figures as you can access. I think that's about it. I mean, obviously there's a lot to look at and sort through, but this is really just to give you an orientation to what's present in the files that you have access to. Okay, so that closes out this this one folder, Nsd Data. Again, we sort of think of this as, this is a folder that you might want to have handy on your local computer or have handy access to look at a bunch of figures, whereas sort of the large, meaty files of beta weights or the raw data or the preprocessed time series or the 40 gigabyte stimulus file are very hefty, and you probably don't necessarily need to refer to those interactively in your everyday analysis.